Welcome to the ZP Developer Zone. So we like to do this webinar once a week, and it's really just answering technical questions that have come in during the week. So I'm going to jump straight into it because we've got plenty of material to get through this week. So it says the 3rd of November here, though if you're watching live, you'll probably realize it's actually the 2nd of November, but I've just gone early because there's so much material to do. I just want to go forward basically. So let's do it. All right, question number one. I'm really going to talk about a paper. Somebody's asked me how they would commercialize or translate a paper like this actually into a product. So the paper is talks about gold nanoparticles on multi-wall carbon nanotubes, and it's a early diagnosis for prostate cancer, and it's PSA, prostate specific antigen. So I will talk about that, and it won't all be talked this evening. In fact, I have the Sensio platform in front of me. The Sensio platform is a super, let's say, easy way of actually commercializing electrochemical assays. So I will, when I'm answering the question, how do we commercialize something like this, I will be discussing this shortly using the Sensio platform. So just hang on for that, if I can put it that way. So that's going to be question number one. Question number two is somebody's and Alzheimer's is interesting because once upon a time the kind of diagnosis for Alzheimer's, it was an unfortunate diagnosis because there wasn't really a treatment. Well, to be fair, now there actually are treatments now for Alzheimer's. I mean, I think as we know, the treatments for Alzheimer's these days are they're not able to reverse the disease, but they certainly are able to by a few months slow up the disease. So I will talk about, and I think there will be a growing interest actually in the diagnosis of Alzheimer's. So I will talk about how to translate a paper like that also into an electrochemical sort of product that you could otherwise sell on the basically sell. So that will also involve the Sensio platform as well. So the Sensio platform is both a vehicle for doing sort of standard, it's like a standard potentiostat. You can do amperometry, it's different open circuit potentiometry, impedance spectroscopy, linear sweep, cyclic, square wave voltammetry. It's different because you can actually drive this using QR codes. So rather than using I've just scanned testosterone, and that's actually going to appear in a minute with one of the hormone sensors. So the app is driven by QR codes. The app is connected in this particular case to the Sensio platform, and through Bluetooth, that is, the connection is, and that will drive what the device is actually doing. I do have some electrodes in front of me. There'll be a question later on about kind of commercialization of things, and I think the easiest way to commercialize actually is to use platforms like this. This is how we can produce really nice disposables for electrochemical assays. But anyway, that will come up in the conversation in a bit. Like I say, I'll go quickly because there is plenty to discuss this evening. So there'll be a couple of comments about how to commercialize these particular assays. It's probably worth saying that questioner number 9 is all about aptamers. But by reviewing questions 1 and 2, you'll also get some insight for questioner number 9 as well. Somebody's interested in drugs of abuse, so I'll do a little bit of discussion on fentanyl as well, which is one of the sort of drugs of abuse that about ZP we do have a lot of experience on. Somebody else is also asking about hormone sensors. In fact, at this moment, I've got this app in a mode for measuring testosterone. So at ZP, we have most experience on testosterone, then followed by cortisol. With the cortisol, we're still the testosterone, I would say, we've got a few partners who are using the testosterone sensor, in particular in saliva. So we are actually shipping a hardware at this moment so that they can start doing that testing. But they're paying partners. And what I mean by that is we're not giving them a technology that we say, oh yeah, this is good to go, you can start using it with end clients. No, they're also sort of helping in the development of that. But in helping in the development of that, they are paying in the helping the development of that. But what they're getting is essentially testosterone sensors that basically look like this, and they've also got the meter, and then we've got the app as well, which kind of drives the assay, and the cloud, the part that where the app, which in fact today I'm not going to talk too much about where the app the app is actually already launched. When I say the app, it's the Sensi All app. It's already launched on the Apple Play Store, sorry, the Apple App Store, and also the Google Play Store. Anyway, this will be something I will discuss a little bit today as well when it comes to hormone sensors. Last week we did a bit of a discussion on vanadium flow batteries. The questioner came back with a few more questions, so I will answer those today. And then we've got a client who's interested in making... I realise I'm answering the question on pH, chloride and sulfate, but they also want to do conductivity. So I'll probably not do a great job on answering conductivity, but the fact that this little Sensi All potentiostat has a mode called EIS, electrochemical impedance spectroscopy. Conductivity in the end is you apply an alternating voltage to a sample, and whether it's highly conductive or not highly conductive then influences the outcoming current. And we do have impedance as one of the methods here. The other ones that these people wanted to do are things like pH, chloride and sulfate, and those are quite easy. So I'll talk about OCP, open circuit potentiometry, which is one of the modes of this particular device. Keep getting amperometry here. Yeah, open circuit potentiometry. And many ion selective electrodes, or many ion electrodes are using OCP as the methodology, but I'll discuss that in question number 6. I realise I need to kind of share the screen a little bit, which I will now do. So I'll maybe just back up a little bit. So I'll talk question number 1 and question number 2 are really kind of sort of binding assays, and I'll talk about how to commercialize those. Question number 3 is really how to detect a drug of abuse. I'll focus in on fentanyl. Question number 4 is about hormone detection. I'll mention testosterone, mostly because in fact that's the one we've got the most expertise in when it comes to measurement in saliva. We do have a few clients at the moment who are busy being kind of, they're paying for it, but they're essentially being beta testers, because the ZP business model is to do contract development, contract manufacturing for other companies. Sometimes we will have quite a platform already built, but then other companies will essentially take those assays to the market, and that's basically our business model. And I will jump to question number 7, which is about lactate sensors. At ZP we do have a lot of experience. In fact, we have a lot of experience in lactate sensing when I think about it. We've got lactate programs on at the moment. Lots of people are interested in measuring lactate in, sometimes it's strictly speaking medical, and then sometimes it's kind of sports applications are quite popular as well. Question number 8 is about ketone sensors, so I will answer a little bit on ketone sensors. I'll give what I can give, which is in the public domain. And then question number 9 is about Aptamer sensors. Somebody struggling to make a Aptamer sensor where they've got some thiol aptamers. They've actually got disulfide aptamers, and they want to put these onto gold electrodes, I suspect, or they are, and turn them into biosensors. So I will discuss that as well. With that said, going forward, we do, if you're interested in doing point of care diagnostics, we do a webinar every month for people in particular who are interested in making point of care kind of diagnostics, like this kind of device, using electrodes like this, and we will discuss that. This is not just a potentiostat. This is actually a potentiostat that, as I say, can do amperometry, open circuit potentiometry, impedance spectroscopy, linear sweep voltammetry, cyclic voltammetry, and square wave voltammetry. This will all become apparent, and in fact I'll probably end up using all of these techniques apart from amperometry, which also is relevant because of ketone sensors and lactate sensors, but everything will get discussed today. But because we've got a data pipe built into this system, we can actually turn essentially raw signals using any of these methods actually into final analyte concentration. And somebody asked a very smart question the other day. It's like, even if you've got let's say you have a nitrate sensor, in the end, the QR code that would drive a nitrate sensor for soil would be different from the QR code that would drive a nitrate sensor for water. So that people understand that actually different matrices will end up in the end with a sort of with different calibration factors associated with them. with that said, we do a webinar every month, particularly interesting to people who are doing point of care or point of need analysis. So the next one's on the 25th of November, one in the morning, one in the afternoon, because that way we cover most of the globe. Wearable biosensors, so if you're particularly interested in lactate, ketones, etc., then come to our wearables webinar on the 5th of November, which is actually just next week. Don't worry if you're watching this later on, these links get updated with the upcoming webinar. In particular, if you're interested in the translation of academic research, I think a few times today, at least on questioner number 9, they're having a problem with reproducibility, and that's really covered in our academic webinar. But I'll put it simply that a lot of the screen printed electrodes that hit the market are fundamentally not reproducible. And that's actually holding up a lot of academic research. That is something that I will cover today. Well, actually, I'll cover that in the webinar on the 10th of December. But the reason that you see us able to develop assays quite quickly is because, in fact, we use our own platform in our own product. So this is something that we will actually ship as a product to end users in the food industry, for example. Just scan that QR code there. Actually, that's ginger. That's chili. So we will ship this as a product ourselves. We use it as an instrument ourselves. And I think the academic, really what ZP is a lot about is actually the translation of academic research actually into products. So if you're doing academic research and you're interested in actually not having your research finish at TRL3, which is technology readiness level number 3, but you want to get it into a more kind of meaningful higher technology readiness level, then please come to that webinar on the 10th of December. Right. Just FYI, a few people have asked me about, well, can you give me a sort of description of how we actually get to market? It's really hard actually to give you just a quick introduction to that topic. I mean, people come to us, they pay us to do roadmaps and Gantt charts and plans on how to actually get to market. And those kind of programs, when we're actually laying out how to get to market, that would really be at least 100 hours worth of effort. But if you're interested in working with ZP in the future, maybe actually you could come to our workshop in Norway on wearables and point of need testing. It's in Norway. It's on the 20th and 21st of November 2024. And again, any links that you see in this webinar, they'll also be underneath this video as well. So this first question is really about translation of an academic paper into a into a point of care device. So one of the problems that we're trying to solve at ZP is there are lots of electrochemical assay type papers produced per year. And in fact, somebody told me from the University or the Metropolitan University of Manchester that there's something like 25,000 papers produced per annum. So that's a lot of papers. And so at ZP, we're kind of keen for more of those papers to actually become products. Why? It basically just grows the entire market for electrochemical biosensors. So the more products on the market, the bigger the market, the more investment will come into the market. So it's basically the kind of, the more we can kind of fill this world with biosensors that add value to society, then the more society will actually invest in it. So it's in our interest to get more people to market. And that's in point why we've actually done the Sensi All platform. So, this exists because there are too many potentiostats out on the market that actually are just serving to do R&D. But this is a potentiostat that actually serves, the fact that we can do linear sweep voltammetry on it and we can do actual assays on it is a very powerful tool. So anyway, back to the point about the translation of this particular paper. So I just want to make sure that you understand that some of the illustrations and data are from this paper, and this is the particular paper. Gold nanoparticles, multi-wall carbon nanotubes based Aptamers for the early diagnosis of prostate cancer, probably through PSA. And I will discuss this. Now, this is a sort of these. Unfortunately, this paper didn't use our screen printed electrodes. It's not unfortunate for them, it's unfortunate for me. But it doesn't mean that people understand that something very different about the ZP screen printed electrodes is actually the reproducibility of our ZP screen printed electrodes. So people think they're a good platform on which they can actually commercialize work like this. So I had to, so I reviewed the paper. In this particular case, we have something called the 501 electrodes, and I think the carbon 501s, which would be a good platform for the translation of this particular paper. Now, if I read this paper, the first thing they're doing is they're really casting onto this electrode nanoparticles, I think it was, and in the end they'll end up it's multi-wall carbon nanotubes with gold nanoparticles on it, and then they'll end up putting aptamers on the surface using a kind of classic sulfur gold bonds, sort of self-assembled monolayer SAM type science. I think they use a molecule called MCH to help. So the aptamers, if you just have a pure surface of aptamers, they won't pack properly. But if you then put some spacing material between the aptamers, which is these MCH, then you can kind of spread out the aptamers a little bit better and get a better sort of packing surface. Then what they're doing then is actually doing that assay. Once they've got their sort of surface functionalized, and I'll describe that, then they're actually doing the assay itself by adding PSA, and they're reading that binding using a sort of Faradaic EIS type reading, where they're using something like ferrocyanide, which wants to oxidize on the surface, but depending on the amount of blocking or binding, I should say, between the aptamer and the PSA, then the signal changes according to it. So it's basically a blocking assay. Now, in this particular paper, this is kind of classically done, that they're actually following their functionalization using kind of cyclic voltammetry. So they build up layers. In fact, if anything, the sensor gets slightly more sensitive. Well, the electrode gets more sensitive as they put their multi-wall carbon nanotubes and their gold nanoparticles onto the electrode. And then as they build up the sort of biological layers, the cyclic voltammetry gets a little bit more distorted, and that's expected because essentially these macromolecules, as they start to coat the surface, are starting to block that surface. Not terribly, but they are doing it. As I say, they didn't use, in that particular case, our instruments or our screen printed electrodes, but if somebody was looking to translate that kind of research into when I say more impactful, I only mean impactful in the terms of, if they're using the potentiostat from ZP, then they actually have a instrument that's not just a potentiostat, because I'll do a demonstration in a bit, but it has something called a data pipe built into it. So in fact, we can turn cyclic voltammetry data, EIS data, square wave voltammetry into actual concentration using a calibration feature that we can actually put into the cloud. And I realized something, actually. I'm going to have to open up something called Julie, but that's something I'll do in a minute. They were following their functionalization also using EIS. So EIS, electrochemical impedance spectroscopy, which is also one of the modes of operation that we have on this particular device. Where's EIS? EIS is one of the QR codes. In fact, I'll just scan it now. Yeah, there we are. Impedance spectroscopy is also one of the modes that we have on this particular device as well. So they're following the functionalization using cyclic voltammetry, and then just to kind of confirm it, EIS as well. But the EIS and the cyclic voltammetry are actually telling them the same thing. So we can use the Sensi All platform for both the kind of functionalization of these electrodes, as they did here, but also then in the actual testing as well. And as I say, they functionalize the surface with an aptamer. The aptamer binds to the PSA, and then they're doing I think they use square wave voltammetry. If they use differential pulse voltammetry, yeah, they use DPV, differential pulse voltammetry. I think differential pulse voltammetry and square wave voltammetry are fairly equivalent as far as I'm concerned. So I do tend to encourage square wave voltammetry just because I find it slightly easier to support people, because I wouldn't say I understand it better. I mean, I understand both of them, but square wave is just a personal preference. It's, there's not much more in it than that, really. In this particular case, the signal goes down as they bind, which is not unsurprising because they're essentially putting a large macromolecule, which is PSA in this particular case, on the surface in a specific manner. They're blocking the ferrocyanide, and the signal is therefore decreasing. And as I say, I mean, they use differential pulse voltammetry, but you can use the Sensei-All platform in a square wave voltammetry and do exactly the same, or generate exactly the same data. And as I say, there's something kind of neat about this particular device that would then allow them to actually go on from just that paper. And I might show that now, if you don't mind. So let me do that. So let me just go into a particular mode of operation. So as I say, this is a kind of a neat device because we're able to go both, we're able to run both as a potentiostat, but also as a sort of, also as a like a final instrument as well. So let me give you an example of what I mean by that. So I'll just take a sample now. I'll put it on the sensor, which I will do. I'll just give this a quick name. Tab. That'll do. I'm just working upside down, so I'm just, excuse me while I am, and I think that's done. And I'll just go continue, and I will go start measurement, and off it goes. So on the screen at the moment, it's actually grabbing it's showing you the current as a function of time, as we're basically just doing a voltammetric sweep in this particular case, and then it will basically process the data and actually sends the data up to the cloud. Now I realize I don't have my browser open, but what I'll do is I'll give you a full kind of demonstration on this. So let me just share my screen with you. Let me just go, I've forgotten exactly what I'm looking for. Yeah, that's right, Julie, good. Let me just go, just sign in for a sec. So what we have is I get a result on the phone, but understand this is actually quite a sophisticated system because it's a potentiostat that is immediately cloud enabled. So I'm never asking the question, where is my data? I am never asking that question because in fact I know exactly where my data is, because in fact all my data automatically goes to the cloud. I can find my particular device, I can find the data that I just recorded, and this is the bit that makes this a bit special. I did a cyclovoltammogram on this, and in fact I've got a result on my phone. So micromolar here is 995, so I've got on my phone the result 995. But also you'll see that 995 is in the cloud. This is the bit that really turns academic research into actual products, because in this system, even though we've recorded cyclovoltammetry and I've got all the cyclovoltammetry in the cloud, I've actually processed the data. So we have something called a data pipe in here, where in fact we can choose to either analyze the first scan or the second scan. I'm just going to turn something off for a second just so you can kind of see that properly. So I've analyzed the second scan, and I've filtered on the data, I've extracted, I've told it to go and look around 250 millivolts, I've told it to fit it to a calibration curve, and I've told it to display the results. This is the bit that makes this very special, that rather than somebody being left with a paper where in the paper it shows a calibration curve and raw signal, this potentiostat will actually take that raw signal, put it through the calibration that you'd already done anyway, because that was part of why you did the paper or when you were doing the paper, and actually turn it into concentration. So now you can understand that, in fact, rather than giving somebody, let's say you said, all right, I want to turn this PSA paper now into a medical product, you would imagine that that gap was actually quite large. But you can generally see that, in fact, if that paper was done essentially on this platform, they would do the differential pulse. What I had actually encouraged them to do square wave voltammetry, because that's what we have. They would do a square wave voltammetry. It would send the square wave voltammetry to the Julie database. The Julie database would find, for example, their peak. It would find their peak height. They would tell it where to look. They would fit it to a formula, and lo and behold, it would actually come back, and rather than giving peak height, it would give a concentration depending on what units they'd actually chosen. And that's something that we've been working on at ZP for 10 years, is how to translate exactly papers like that into actual products. Why? Because the more translation of academic research into electrochemical products, the bigger the market, and it's better for both the academic community, but it's also, it's also very good for commercial organizations like Zimmer and Peacock as well. Otherwise, the big problem I think that's been going on in electrochemistry for decades is everyone's using potentiostats that are designed to do R&D, and the Sensei-All platform is the first time that anyone's actually made a potentiostat that was genuinely actually trying to be a real product. I mean, just alone, if I take one of these screen printed electrodes, put it in there, snap, that's a user experience that, that's the kind of details that users like nurses and doctors and clinicians, etc., are actually looking for. Okay, so that was a quick demonstration of that, but what you saw there was I used the Sensei-All potentiostat. We used one of our screen printed electrodes. We put the sample on there. I scanned the QR code on my phone. The QR code, or the phone rather, is in Bluetooth connection with the device. We told the device what to do. The device did the assay, sent the data back to the app. The app sent the data to the cloud. We processed it in a system called Julie, and we actually went through pre-processing, processing, and information, and we actually turned it into final concentration. So I hope that was a useful way of understanding. And we've got lots of these types of cartridges as well. So at ZP, it wasn't my intention, but I realize now that, in fact, if we start to produce our screen printed electrodes like this is a product. This is what you can actually give to non-electrochemists. And when you give screen printed electrodes, it's very hard for people actually outside of electrochemists to do, outside of a lab, to actually handle them. But what we have is a very different form factor, and it's no more like awkward slots where you're trying to slot this screen printed electrode into the device. I mean, what we have here is a very elegant way of just, place the sensors in there, or the screen printed electrode, snap it shut, and it's done. There's all sorts of user experiences with this. There's like the LEDs on the front, etc., that really make this easy, and the fact that it works with a phone, and the phone is just automatically connected to the cloud, really just help with the user experience and also with the data. Now, question number 2. I think there's going to be a certain surge in people's interest in actually doing diagnosis of Alzheimer's. Like I said, I remember, I know that this kind of work's been going on for years, and I was kind of like a bit concerned because I was thinking, well, you can have all this diagnosis, but if there's no treatment, what's the point? Well, what's good is circumstances have overtaken that opinion now, because in fact there is a treatment. You can't get it on the NHS in the UK, and they consider it too expensive, but the drugs will get better and the treatment regimes will get better, and then that return on investment by the things like the NHS will change and it will become available. But I suspect in the US people will demand it and they will get it. So if I read a paper like this, they were particularly using glassy carbon electrodes. I like glassy carbon electrodes, but they just do not translate into products. So if you want to translate into products, then you have to search yourself out a good carbon-based or graphite-based screen printed electrodes. I know people are very keen on graphene these days. I really think that graphene doesn't add a lot of value to assays. In fact, I think it can actually be quite an expensive material. I'm sure the cost will come down, but graphene is not this wonder material. The reason that people would use graphene is because it seems novel and therefore you can publish with it. But when you're really trying to translate science into products, whether it's publishable or not is not here or there. Actually, what people are looking for is low cost, easy to use medical diagnostics. So, graphene doesn't add a lot of value in that particular case. With that said, if we're going to translate this paper into a product, then I think the 501 screen printed electrodes from ZP will work. Having read the paper, we've got these carbon nanotubes, we've got this chitosan, we've got these gold nanoparticles, and essentially these get deposited, in their particular case, onto a glassy carbon electrode. But as I say, you can't commercialize off a glassy carbon electrode. So in fact, what we would do is drop cast them onto a screen printed carbon electrode, and then the gold nanoparticles can basically be used to anchor the antibodies through a thiol atom or sulfur atom, and then they're blocking to stop non-specific binding using BSA, bovine serum albumin. Again, you're able to follow this kind of functionalization, and I did, again, this material is out of their paper, so I just want you to know that, and I'm only reviewing it in order to think about how to translate it. when you read a paper like this, actually what you find out is when you put the carbon nanotubes down, and what they're doing is they're doing functional steps and then checking it with sort of ferrocyanide. So they've got their kind of pristine glassy carbon electrode, and then when they essentially functionalize it with their carbon nanotubes, the sensitivity goes up a little bit, and they're basically just kind of adding extra roughness to the surface with these carbon nanotubes, or extra surface area. So I do understand that. And this kind of cycle voltammetry they use, or they're also using differential pulse voltammetry, though I would probably tend to use square wave voltammetry. It makes a little difference. Again, we can use the Sensei-All to both do the functionalization and the characterization as they do in the paper. Having understood that, they've got this functional, they've taken essentially a carbon electrode, and they functionalize it with the chitosan, the gold nanoparticles, the multi-wall carbon nanotubes. They've blocked it with BSA, and they've put an antibody in this particular case onto that surface. And then when it binds, it's essentially blocking the surface, and the amount of binding with the target, the greater the greater the blocking. And so when they've got sort of 10 picograms per mil to 100 picograms per mil, the signal is decreasing because essentially it's a blocking assay. But that is definitely something that will translate well on the ZP Sensei-All particular platform. Question number 3 is, when people ask me questions about things like drugs and abuse, then I'll try and bring in knowledge where we have first-hand knowledge. So at ZP, we actually do have first-hand knowledge. Again, this kind of work, we will have done it on our Sensei-All platform, using our 501 carbon electrodes. And here we've got fentanyl and morphine. This is the 0 micrograms per mil. And then when we detect these molecules. Both the blue one is the fentanyl, and the red one is the morphine. So they give different signals, different peak positions, and also different intensities as well. So with this kind of work, we do have that kind of experience. In this particular case, ZP will do contract development, contract manufacturing, but ZP's contract development and contract manufacturing works best with well-funded people. So, unless you're really able to kind of, fund into the hundreds of thousands, eventually into the millions, and if you're based in academia, then sometimes it's better to actually do the work yourselves, because, your labor costs are under your control. When you try to use an ISO 13485, which we are, so Zimmer-Pickel is an ISO 134 contract development, contract manufacturer of electrochemical assays and biosensors, and things like wearables. I mean, I've got a wearable system in front of me as well. So I'm talking a lot about point-of-care diagnostics. This is a particular wearable biosensor that we also have as well, also using electrochemistry. And I bring that up and just say that if you are in academia and you're looking to make sensors for maybe detecting some of these drugs of abuse in things like blood, I'm not sure that ZP is necessarily the best vehicle, because our cost of development is quite high, and so we work well with either large companies or well-funded startups. People can try us in academia, but I just don't think the budgets are there. Otherwise, you would have to sort of essentially do the work yourselves, and if you're going to do the work yourselves, then you will need probably carbon electrodes of ZP and the Sensei-All platform, and then you essentially have to start doing the investigation yourself, mostly looking at the literature and finding what prior art there is on that type of target. But it is something that we have done at ZP, and we do have experience of it. Again, so the nice thing about actually using the Sensei-All platform, I would argue, is that actually it is essentially, it can both do lab work, and it can be, let's say, a minimally viable prototype that you could take out of the lab and start using in a clinical setting as well. So question number 4 is about somebody's interested in testosterone, which we've got direct experience in. Estradiol, we don't have direct experience in it. Prolactin, we don't. Cortisol, we definitely do have direct experience in. I mean, if we can do, the reason we haven't got down to estradiol and prolactin. It's just we haven't had a commission off somebody. So some of ZP's income is from contract development, contract manufacturing. For example, this watch I'm wearing came about because we worked for 5 years with a company called PK Vitality that was making a microneedles for continuously monitoring of glucose. So that's kind of one revenue stream for us. And then we also do a lot of more like point of care diagnostic development, which when I say point of care diagnostic development, that's a kind of an example of a point of care type diagnostic, even though it's also a potentiostat as well. So we do have experience in testosterone and cortisol, but nobody's really commissioned us to do estradiol or prolactin yet. At this moment, we are shipping to a particular client. When I say client, the kind of we very rarely face the end user. We're facing other businesses that are otherwise trying to get to market. So at this moment, we are shipping the Sensi-All platform. So the Sensi-All platform actually does have a testosterone assay on it. So in fact, this app, if you're not clear about this app, maybe come to the webinar on the 5th of November. You'll find a link underneath this video where, in fact, we may discuss that a little bit more. But we are shipping units like this with the app that you can download off the App Store, along with sensors to clients at the moment. And the clients are helping to validate the measurement of testosterone in saliva. So that is definitely something that is going on. So we do have experience on that. Now, the sensitivity and specificity, we've obviously built this using biological molecules that are inherently specific to the testosterone, and the sensitivity is in the clinical range because we've done our due diligence, and we've what do we expect in saliva, and therefore tested in those ranges. It's hard. Can you outline the stages for the development process from prototyping to full production? Well, I presume the nice thing about ZP is we've really accelerated the prototyping phase. Now, what I mean by that is, here's a prototype. It's even got a testosterone assay built onto it. We even have testosterone sensors. So the prototyping is very much accelerated with ZP. In part, the reason ZP, one of the things everyone talks about, I don't want to reinvent the wheel. If it's been done before, I want to do it. And everyone reinvents the wheel. But at ZP, we realized that, so ZP started 10 years ago. Most companies that we would have seen in those days have already hit the wall. Most companies, go out of business in 2 years, 5 years, 10 years. Some of them scramble on for 15 years. But there's in the end, very few people are actually successful. And in part, it's because actually they spend so much time trying to get to this stage that they're not really working on the value add. The value add is doing the business development, finding customers, finding out what the customers really want, finding out if you can really sell this to actual customers. And they spend a lot of time actually doing the science and engineering. And so at ZP, we've tried to build a platform upon which we can really accelerate that prototyping stage. So, if somebody came to us and said, I want to demonstrate in front of some investors, an MVP, a minimum viable prototype, I'm being very careful with that use of P there, then, we're essentially good to go on that. So the prototyping stage is very much accelerated with ZP. Full production, well, to be fair, actually, now I know I'm showing a single channel device here, though I would want you to understand that, so for example, this is already a multi-analyte system. So if I wanted to measure potassium, for example, every time we create a new QR code, I've now got potassium back to testosterone. So now I'm back to testosterone. Let's say this is saliva. So let's say I want to do pH. I'll come down and scan a QR code for pH. So we got pH there as well. So it's actually multi-analyte. It's just that we do have to have different sensors for different analytes and different QR codes to drive different methodologies on that particular platform. What happens is people say, people will actually give themselves quite high engineering barriers, because it's nice to have engineering and science barriers because those are the kind of things that we're trained to do. But really, you actually want to find out what's the minimum feature requirement I need to actually really test out whether I'm actually going to be able to sell this. And so just a single channel on testosterone is probably a good place enough to start. So we are actually in full production on this particular device anyway. The business model of ZP is actually not to, it's the really cool, it's called OEM, so be an original equipment manufacturer and let other people actually take some of this technology to market. So we can already manufacture at pilot and full scale. We're already, so if you go to the ZP website, you can buy sometimes pharma products in tens, or you can buy, for example, we can manufacture in millions. Let me just grab an example here. So here's a sheet of potential testosterone sensors. There's 300 on that sheet. We can produce thousands of these type sheets easily in a week, but hundreds in a day. So our capacity for production is much higher than actually you could necessarily sell. Minimum, really, what it comes down to is to really engage with ZP, you need to have the correct funding. You need to be able to work on multi-month, multi-year programs ZP. So it really just comes down to funding. It's hard to work in stop-start mode. Every month something has to be happening. So we are looking for people who are particularly well funded. For example, if you look at the history of IVD, in vitro diagnostics, I should say point of care diagnostics, not in vitro diagnostics, the most tens of millions have been spent by these companies to actually get to market. I think actually that spend is not necessarily particularly efficient, but I would say that if you really were going to go to market with a particular product and get, if it was FDA approval, then you're looking for at least 5 to 10 million US dollars to really do it properly. I don't think you need to sacrifice on performance. Actually, cost and performance. In the end, at ZP, what we're doing is we're repositioning glucose strip technology into new markets. So I don't think there actually necessarily is a compromise as the way you put it. Especially when you think about testosterone testing in saliva, what is your competition? Who are you being compared against? And the answer is not much, actually, if anything, at this moment in time. So I don't think there's necessarily a compromise to be made here. You do ask an interesting question at the end here. So let me answer this question, right? Which is, how are we going to do this? Are we going to do this by optical, or are we going to do this by electrochemical technology? So let me just change cameras for a second. Now, obviously at ZP, I'm going to give you a completely biased answer. You see these? So these are glucose strips. Now, this is the kind of technology at ZP that we're based upon. We use this kind of technology quite heavily. What does the glucose strip teach us? It teaches us that if I want to have a complex sample, like blood or saliva is also quite a complex sample. So I got some blood here. I do that. 5 seconds later, I'll get the result. This technology is electrochemical because actually it's easy to manufacture. It can be low cost. The electronics are low cost. The electronics are small. And in fact, electrochemistry can be fast. So I've got a glucose reading here of 113 mg per dl. 90 mg per dl is pretty standard. This technology didn't start off electrochemistry. In fact, the first generation of at-home glucose sensors were optical. But with optical, you immediately run into especially with things like saliva, you immediately run into that optical systems often need a transparency in the sample. So you have to probably do a dilution. And once you start getting into dilution, you're diluting the actual concentration of the analyte itself. You have to prepare it. So a lesson from history. Nobody wanted electrochemical sensors for glucose sensing. They only went to it because the low cost of manufacture like this. In fact, when you saw me do that test, there was no sample preparation. And you also notice that this is sitting in ambient light. And so actually that makes for a low cost meter. So in fact, electrochemistry basically won through in the end and completely wiped the optical systems for at-home diagnostics off the market overnight. So don't misstep if you want to make a low cost point of care or consumable, point of care diagnostic, and it's for at home use, don't misstep and start on optical, because you've got to learn a lesson from history, and the lesson from history can be learned from the glucose market. Electrochemistry wasn't the first technology that actually was used at home in glucose detection. It was actually optical. But once the first electrochemical strip came along, it was wiped out. So if you go to market with a optical testosterone sensor, I can assure you that somebody will come along with an electrochemical one and essentially wipe you out. So don't misstep and learn something from history. I mean, post-development support, at ZP, we're an ISO 13485 contract developer, contract manufacturer. So in the end, if somebody goes to market, we can be their contract developer, contract manufacturer when they're on the market. Question number 5. So we do a lot of, support, and we don't, this support, we're not expecting remuneration here. And this would be one of those kind of cases where somebody is a student, they're interested in vanadium flow batteries. I'm kind of quite interested in vanadium flow batteries as well. Just a really geeky kind of interest. I quite like power and energy. I do like renewable power and energy, and if you're going to do this, we're going to have to store energy production when the energy production is happening, but the consumption's not there. We're going to have to store that power, and vanadium flow batteries is just one way of doing it. I did do a webinar last week about it, where vanadium 4 plus is oxidized to something like vanadium 5 plus, and then on the other side, vanadium 3 plus is reduced to vanadium 2 plus. Then the questioner came back and said, well, how do I get hold of vanadium 4 plus? How do I get hold of vanadium 3 plus? I only did a quick Google search, but I just went and had a look, and you can buy vanadium 3 plus. Here's vanadium chloride. So that's an example of vanadium 3 plus, and you can buy vanadium 4 plus. Vanadium oxide sulfate would be an example of vanadium 4 plus. So the quick answer to your question is, this is just a public service from us to you, but you can basically buy these materials. So there's your answer. Ion selective electrodes. So we have a particular client. They're interested in developing sensors for quite a complex matrix. So what I say now it will definitely guide you, but understand you've still got a lot of science to happen. At ZP, we're actually really expanding our availability of these kind of cartridge screen printed electrodes. And I think in particular we've got a carbon version, which I think will be particularly useful to you, this carbon 501. So if you're interested, and I know you asked about conductivity, and I forgot to answer conductivity, but the quick answer is this particular electrode, it's called the hypervalue cartridge electrode. It's carbon in particular, but would probably work quite well as a conductivity sensor if you ran in the EIS mode. So I think you have ordered a Sensor or potentiostat. You've got EIS in there already. You may have even ordered some hypervalue carbon electrodes in the cartridge mode from us, and if you have, then that will probably do your EIS, sorry, not your EIS, your conductivity question. Your other questions then were about you were interested in chloride. So a lot of anions can be detected using ion selectivity. On the ZP Sensor or potentiostat, we have open circuit potentiometry, so that's the mode of operation that you need. It's a really straightforward mode of operation. If I scan my QR codes, there's only runtime and save rate that I have to mess around with, so it's super simple. Just FYI, if anyone's not so familiar with electrochemical techniques, again, we are having that in-person workshop at the end of November I think it's the 20th and 21st of November. But reach out to us if you think you've missed it, because there will be other workshops as well. Anyway, back to the point. So for chloride, there's an ionophore exists. I'll expand on this in a minute, but I just went on. I used to call it the Sigma Aldrich, but Merck have bought Sigma Aldrich. So there's a chloride ionophore, and there's a pH ionophore or hydrogen ionophore, and there's a sulfate ionophore. So I haven't deeply looked into this. I'm only giving a hint, but I think if you were to search up open circuit potentiometry, ionophore, the anion of interest that you're interested in, you will find that actually there are formulations out there, and you can buy these ionophores. And I think what happens is you take the ionophore of interest, you probably find that the papers say to dissolve it into THF, tetrahydrofuran, and then you essentially drop cast it onto hopefully a ZP screen printer electrode. And then THF is quite volatile. It's boils at about 60 degrees C, so do it in a fume hood. But it should evaporate quite quickly and leave you with the ionophore. I realise I've left out something here, which is you probably put it in PVC as well. You make a blend of the ionophore, PVC, polyvinyl chloride, and the THF, and that's what you drop cast, because then you're left with this kind of PVC with these ionophores embedded in it. Have to read the literature, but ionophores are available. They're available for all the anions you're interested in. THF is probably the carrier solvent. I think PVC, polyvinyl chloride, would be your sort of polymer. So you're essentially making a ion selective polymer, and you'll probably do drop casting onto a ZP electrode, and you would measure it using open circuit potentiometry. Right, lactate sensors. All right, so we're very active in lactate sensors at the moment. It's funny actually, lactate, ketones, and glucose. So glucose is really proven out. Glucose wearable biosensors have really proven out that there is a viable biosensor wearable market. This is not my product. I have no sync with it, but you will see people walking around with these, and it's a demonstration that people are interested in their metabolic health. And I mean, I know that athletes have been pricking their, basically drawing blood or doing little blood tests for years on their lactate. Okay, let's jump into it a bit more. Can I provide more information on the procedure and pricing for developing and manufacturing such a device? So, I mean, it's a really hard one, right? So, just FYI, you see this wearable watch, which is actually using microneedles. So far on this watch, $20 million has been spent on this watch so far. So that happens to be in the glucose and ketones mode, but we've also done it in lactate as well. But $20 million has gone on that so far. You see this Abbott product. I could easily estimate that Abbott spent $1 billion getting this to market. Wearables are quite tough. Now, if you go after the sports applications, then you can find some softer routes to market. But I would honestly say that in order to make a wearable lactate sensor, you are talking at least $10 million to kind of get to something that's MVP, minimally viable prototype. I would honestly say it's quite a tough challenge. Now, we're actively doing this with clients, and I know how big their programs actually are. In fact, I might have a link at the moment. If you want to take a low-cost start on this, then ZP does have some landing pages where you can kind of buy this kind of technology quite low cost. If people are serious about working with ZP on things like lactate sensors and they want to kind of get a backdoor way to find out a bit more about this kind of stuff, then maybe those workshops that we're doing at the end of November are worth coming to, because then you can kind of meet the team, ask your questions a bit more. But you know, I can give you examples. So I mentioned Abbott probably spent $1 billion developing their technologies. Not probably, it definitely is. I can tell you that Dexcom, who are doing a glucose sensor, which is pretty similar to a lactate sensor, spent $500 million at least getting to market. I can tell you that BioLinker, that's doing glucose, has spent $158 million in the last 4 years doing glucose. I can tell you that this one had $20 million so far spent on it. So why have I said all that? I just wanted you to know how expensive it can be. Now, I think if you partner up with ZP, you're talking more like 10 million, only because then we've actually got so much experience. A lot of spend actually takes place on training and building teams, and teams get fired and replaced, etc. So I actually think that a lot of these costs are all about inefficiency of spend. But with ZP, I think it's actually a lot lower cost, but it's not zero cost. But if you want to go alone, just FYI, that some of these links will take you to, you're able to actually build these kind of sensors on your own. If people have the funding, they can actually come to ZP and do some lactate sensor testing, especially if it's the sports applications. But you really do have to have funding in order to kind of come to us, do some testing, go away with your learning and then maybe launch your businesses after that. But as I say, I think actually one of the lower cost ways of thinking about this is actually just to come to our workshop on the 20th and 21st of November. Ketone sensors. So at ZP, we've been making ketone sensors pre-2019. We've actually been making ketone sensors, but we've actually been making ketone sensors on wearable formats, including microneedles of recent. The data I'm putting here is no different to what's actually in the public domain. But if people are interested in doing ketone sensors, then you can essentially potentially send us substrates. You have to pay for that, sending us substrates to us, but then we can functionalize them actually into ketone sensors, test them, and send them back to you for further testing. So question number 9. Right, I'll take a little bit of time on this. So this is an academic, or somebody doing academic research. They're trying to make an Aptamer sensor. So they're trying to make it for thrombin on a thiol modified gold electrode. In fact, and they are using screen printed gold electrodes. I hope they're not using our screen printed gold electrodes. If you are, I really apologize, because I actually think that we make the world's best repeatable electrodes. But I'm going to talk about your problems. I can understand that you're trying to follow this reaction by impedance spectroscopy. Square wave voltammetry and impedance spectroscopy will both do the job quite well as long as the actual sensor is working. Sometimes you observe no signal change, and sometimes you do. So you've basically got a repeatability or fabrication problem. No problems will deal with that. You get significant fluctuations between electrodes. I really hope you're not using ZP screen printed electrodes, because if you are, I'm embarrassed. I've got a strong suspicion you're not. And that is a big part of the problem, that so many... I'm doing an academic webinar on the 10th of December. Please come to it, because there I'll talk about... There's so many vendors producing so many screen printed electrodes, and actually so few of those people or vendors are actually using these screen printed electrodes in their own work or in their own products. At ZP, we're really different. We use our gold electrodes all of the time in actually developing an Aptamer sensor for a client at the moment. So well before you would ever have a problem with ZP screen printed electrodes, we would know about it. So we don't ship bad screen printed electrodes. It's just different the way we actually work. So you say between electrodes, fundamentally the electrodes you're using are not repeatable. That is where your problem starts. I'll talk about controlling variables. Some of those variables, by the way, are outside your control. Because if the electrode if the electrodes you're using are variable, that variable is outside your control. And then I'll talk about how to reduce other variables. You ask whether you should, these aptamers come as a disulfide. Should you actually electrochemically reduce the disulfide into thiols? And I'm going to say yes, you should. Because otherwise it's actually a variable that's outside of your control. But when you deliberately do the reduction of disulfide, you're controlling that process more. I'll give you some hints on time to temperature. You did say ask for here on concentration, but I must admit I haven't answered that bit. You asked a question here whether Faradaic EIS is a little bit outdated. It's kind of an interesting question actually, because the one thing I would say about Faradaic EIS Aptamer sensors, I'm thinking carefully, there are no Faradaic EIS Aptamer sensors on the market actually. So the reason being I bring it up is because you say it's outdated, but maybe in the academic literature, but nobody's actually translated this kind of technology into... I think the people who are closest are actually ZP with our clients, but nobody's actually, as far as I know, done it. Genmark DX is the closest, but they do something different. So uncontrolled variable is your screen printed electrode. I strongly suspect you don't use ZP screen printed electrodes. That's not your particular fault. But at ZP we're quite different. I said the first people to know whether a ZP screen printed electrode is, especially a gold one, is functional or not, is ZP, because in fact we test them before we release them to ourselves and before we release them to clients. So, we'll make them in quite large sheets like this, and we'll actually sacrifice quite a few per sheet to actually make sure that the sheet is generally good. And something that we really aim for is repeatability. So you have said it in your question. I think the biggest problem you possibly have is repeatability. So what do? We test the peak heights. We look at the standard deviation of the peak heights. We look at the relative standard deviation of the peak heights. And we're looking for an RSD that's in single digits. I think a lot of screen printed electrodes, if you took 10 or 20 of them and tested them all with ferricyanide, they'd give you quite different results. And that would really, really mess with us at ZP and also mess with our well-paying clients. So we can't have that. And the reason we're so fast and efficient at actually doing assay development is because the materials that we're building upon, our screen printed electrodes, are so reproducible. So your biggest problem is actually reproducibility, and not all these screen printed electrodes will all look beautiful from all these various vendors, but how many of those vendors are really using these screen printed electrodes to actually do commercialization? And I answer very few, if any, apart from ZP. So we do a lot of work on making sure that the electrodes are repeatable. The other thing that may be going on here is that at ZP we're very experienced. When I'm talking about making Aptamer based biosensors, I'm not talking about this in the abstract. We generally do this. So the next problem I suspect you've got is gold is really good at passivating. It's really good at absorbing material from surfaces. So in fact, I have a video in this link. You have to watch that because you actually have to activate that gold before you really start the process. So we do an activation and we rinse it. So even if we've produced a screen printed gold electrode and they've passed our quality assurance, just before we start doing the functionalization, we actually activate them by essentially chemically cleaning them. I didn't see any mention of that in your notes. So we do a chemical cleaning step. That's the first, the second part. First part is we use reproducible gold electrodes. How do we know they're reproducible? Because we do so much quality control on them. Even if we've done that quality control, even before we start the functionalization, we actually activate. But we then don't leave these electrodes hanging around. We immediately use them. You asked the question whether you should just essentially split this disulfide bond using TCEP, and I think the answer is not whether you should, it's that you should. Otherwise, you're kind of got a process where you're kind of relying on that gold to split that bond. So it's slightly out of your control. You should make that more deterministic by actually just putting in this reagent, letting the reagent reduce the disulfide bond, and then actually you should then get a more repeatable surface. We actually use quite a large excess of this reducing agent, something like 50x, even sometimes 100x of this. It's hard to call on this because each aptamer, the ability to reduce it, that sulfide bond can be a little bit sensitive to the actual aptamer itself, but we use quite an excess of material actually to get that reduction. So we don't rely on the gold. I think we've tried doing that, it doesn't work for us, and we definitely do a reduction of the bond. What am I going to say here? So we do characterize every step of our functionalization. Earlier on, there were two papers earlier on that I was talking about the commercialization of question number 1 and question number 2. Go back and watch those videos because it's really important to see how they actually followed, each functional step by cycle voltammetry. I'm sure you're doing the same, but after you've activated the surface and rinsed that gold electrode, you should check it with, we would actually check it with ferrocyanide and just make sure that we got a good CV off it. And you can check whether you've got a good CV by looking at the data that I got previously in these slides. If you've got a good CV, then you've reduced your disulfide to a thiol. You can put that thiol on there. I noticed you didn't mention anything about a spacer, so MCH is a good spacer. You should have that in there as well to keep the molecules, and then you should be quite good to go. Now, what I'm describing is something that we spend. We do hundreds of hours of this kind of work at ZP per month. So what I'm describing is not in the abstract. Let's say, as you add those thiols on, the cycle voltammetry should get distorted because you're essentially blocking the surface. You mentioned annealing. What we would do is we would form this thiol bond. We would form this self-assembled monolayer between the aptamer, have some MCH in there as well as a separating molecule, and we would leave that actually sitting in the cold for about 12 hours to essentially let the surface arrange itself into its lowest energy state. We don't necessarily add heat in order to help it anneal to that. We don't, is the quick answer. And then you should be ready to read it. And by reading it, I just mean, bind to your target, which I think was thrombin, if I remember rightly. And then you can read it by I know you were asking the question, well, is EIS Faradaic a little bit outdated? It's ironic because it could be outdated in the academic literature, which may be all you're targeting, but in terms of commercialization, it's not outdated. In fact, there's very few instruments in the medical space that use EIS. So you can essentially characterize this by cyclic voltammetry. You can use EIS to read the signal. You can use square wave voltammetry to actually read the signal as well. I appreciate you're probably not using... I don't think you're using our screen printed electrodes, and so therefore, and I certainly don't think you're using our potentiostat. But if people are interested in making Aptamer biosensors, then of course, the completely biased perspective from me is use the sensor or potentiostat, because it's both good for actually functionalizing these kind of screen printed electrodes and making them into essentially biosensors, but actually then it also has a very nice capability of actually taking this, taking an assay like in this particular case they would use EIS or square wave voltammetry, putting it through the data pipe and actually turning it into products. So your question was, is Faradaic EIS a little bit outdated? And I would say there's a different spin on this. What's the biggest problem in academia is actually the lack of translation of papers into things that really look like products. People will show kind of homemade devices, but really, they're not really able to take them onto products. So I'd actually say that your paper should be different. That actually you should demonstrate the capability of translating onto a platform like this. So in fact, you can actually demonstrate that your work generally can have impact. Otherwise, it will just be another paper. So question number 1, 2, and 9 are sort of related. So watch those 3 if you're particularly interested in Aptamer type electrodes. So question number 1, we talked about the the translation of a prostate cancer, or PSA, prostate specific antigen assay, into a commercial product. It was very similar to question number 2 actually, and question number 2 is interesting because now there are treatments for Alzheimer's, so actually diagnosing Alzheimer's rather is actually more relevant than ever. They were both using gold in there, which is actually relevant to question number 9. Question number 3, at ZP we have actually done morphine and fentanyl on this sensor or platform. If the questioner is coming from academia, it's really hard that academia can't really justify spending the kind of money with ZP that it needs to develop those kind of assays. But if you want to essentially skip the ZP part, just get the sensor or potentiostat and get some of our carbon cartridges and essentially do the assay development in academia, where the labor costs, etc., are going to be much more reasonable. Hormone detection, we do have experience with testosterone in saliva. We do have clients now receiving our testosterone in saliva, and they are helping to validate that, but you do have to be well funded. Don't go down the optical route. It's really a step backwards. Google the history of glucose strips and find out that history to help you justify or help you understand why I say what I say. Vanadium redox flow batteries, if you're looking for vanadium 4+, vanadium 3+, you can buy them from Merck. So quick answer there. With the modifying for pH, chloride and sulfate, you can get those ionophores. You will end up making a solution of ionophore plus PVC plus THF. Do try and do some literature searching around that, because I haven't actually given you an absolute formulation, but I know those exist. But that would be the strategy. Measure it by open circuit potentiometry, and you probably need some of those 501 cartridges from us. Lactate sensors, I kind of painted that slightly gloomy picture from the glucose world, but I just want people to understand that wearable biosensors is not a small task. You can skip and do some easy prototyping using some of the technologies at ZP, but I don't want to paint this picture that this is going to be super easy, let's say. And if you do want to skip some steps, come to our workshop, meet ZP, understand who we are, see if you can work with us. Ketone sensors, I haven't given a lot more than is already in the public domain, except I've given you a hint that actually we've done it onto quite complicated sample types like microneedles. And then Aptamer sensors, you've got a reproducibility problem. I think it starts with the screen printed electrodes. When I read the details, I don't see a cleaning step in there. When you put the Aptamer down, I don't see you using spacer layers. We don't just put the Aptamers straight down there because they come as the disulfide. You have to split that with TCEP. I think it's the right acronym. I think at that point you should be good. And the papers in question 1 and 2 should be quite informative for you. And I think with that, I want to say thank you very much. I can't even tell you how long I've been talking for, but I would suggest it's probably too long. So with that, I will round up. Wish everyone a good week. If you've got any questions as EP, you can see that we do put quite a bit of effort in, and we understand that some of these questions have no economic benefit to ZP at all, but we just do it because if it's interesting you, then it's fun. Okay, if you've got any questions, I realize I might not even be sharing my screen for some of those answers, so I apologize if that was the case. But if you've got any questions for ZP, don't hesitate to reach out to us. Okay, thanks very much.